Greetings. Thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members in the broader scientific community about topics related to antibody discovery and development. Our expert speaker, Dr. Noel Smith, PhD, will discuss the advantages of integrating a high-sensitivity immunopeptidomics assay in preclinical immunogenicity risk assessment with us. Please note the webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Without further ado, I'll turn the show over to our speaker, Dr. Smith.
Thank you, Kristen, and welcome everyone. Thank you for joining. Before we get started, I just want to apologize. I do have a very heavy cold, so please bear with me if I have to mute my line or to cough or anything. I'll try my best to keep it to a minimum and stay as clear as I can. So, as Kristen said, I'm going to talk about how we integrate a MAPS assay into our preclinical immunogenicity risk assessment platform. I'll talk a little bit more generally initially, and then also show some case studies as we go through the presentation. Just a couple of quick slides just to set the scene. I work at Lonza. I head up the immunology team that we have. I've been working here for just short of 16 years. I'm based in Cambridge in the UK, so the top image you can see here on the slide. And we also recently, about 18 months ago, opened some labs in Cambridge, Massachusetts. So just to confuse everyone, we have two different Cambridge sites. So I have a team across both sites, and pretty much all the assays we're discussing today are run both in the UK and in the US. And my immunosafety group sits within an early development services group. So we work in kind of early stage development, late stage discovery with clients, lots of biotech, pharma, even academic groups, to help really kind of optimize the lead before you go into GMP production and clinical trials. So we're interested in making products as manufacturable as possible, reducing any risks as you go through the GMP process, but also safety, immunosafety in particular, which is obviously the focus of today's talk. So just a quick high-level overview. I'll spend a brief amount of time with a bit of introduction to immunogenicity. I'm sure a lot of people on the call are fairly familiar, but for those of you who aren't, I'll give a basic overview. I'll touch very briefly on regulatory expectations and how those are changing as well across the industry. I'll move a little bit into in silico tools super briefly, and then spend most of the talk in the in vitro assays and the MAPS assay in particular. And then talk about some of the applications for the assays, how we generally apply these sorts of studies, at what stage, with what sorts of molecules, etc. And then probably why most people are here to see the kind of data on a MAPS case study that we have, that we've had quite recently on a really interesting study. And then I'll wrap up with a few conclusions at the end. So just starting with the introduction. So my team, working on developing assays and technologies to look at everything related to immunogenicity assessment. We do a lot of work in the vaccine space as well, so that's where you want an immune response. Immunogenicity is good in that setting. But generally speaking, when we're talking about biotherapeutics, cell and gene therapy, etc., they're unwanted responses. We don't want the patient to raise an immune response against the drug or the modality that we're treating the patient with. And the safety can be further broken down into aspects of the immune response that can lead to anti-drug antibodies. So this is typically the more general immunogenicity that people refer to when they're talking about clinical studies. We're measuring anti-drug antibodies that patients develop against the drug. Normally, they're not associated with safety issues. They clearly can be, and there are some cases of that. But usually, it's more of a detrimental impact on the PK, PD, and hence the efficacy of the drug. So it's definitely something you want to avoid from a drug performance perspective. And there are lots of cases out there that I'm sure most people know where drugs work really well in the clinic and they get immune responses in the patients, and then people have to switch away from that drug because they stop working, essentially. And then in red here, we have the really bad stuff that we really want to avoid, the immunotoxicity events, things like cytokine storms or off-target toxicity. So they're the pure safety events. And we have a number of assays and technologies to look at all aspects of the immune response to therapies. Just a quick note to make a point that immunogenicity is very complex. It's very difficult to predict. As I'm sure all of you working in the industry know, it's not an easy thing to do. It's multifaceted. There's lots of different contributing factors that determine whether a patient raises an immune response against a therapy. And on the right-hand side of the slide, you can see the patient and treatment or sort of more clinical related factors. And they're much more difficult for us to predict and model in silico and in vitro. So they're the things that really have to wait until you go into clinical trials to really fully understand what risk they pose. But what we can do with the in silico and in vitro tools is more so focused on the left-hand side of the slide here, where we can look at aspects of the protein in this example here, where we have the species of origin, T and B cell epitopes, post-translational modifications. All of these play a role, and these are things we can assess the impact of in silico and in vitro. But not just the protein, the sort of API itself. We also have other aspects of the product, like co-cell protein aggregates, excipients, and other product and process-related impurities as well. So we're very much focused on sort of the left-hand side of this slide when we're using these human in vitro preclinical tools. And as I said, I did want to touch very briefly on the regulatory landscape. It is changing pretty rapidly. Most of you will know that for an IND filing, for example, for your first in human application, you will need to provide an immunogenicity risk assessment, but there's no set regulatory guidelines on exactly what that entails. What in silico you do, what sort of in vitro assays, what scale you need to run these on. So you do really need a team of experts to talk to internally and externally, people like myself and my group and other CROs offering these sorts of services, just to really understand what makes sense for each individual program. So we have to take into account the modality, the disease indication, the mode of action. Lots of different things play a role in just how detailed an immunogenicity risk assessment makes sense for that individual program. And as I'm sure most of you know, obviously a lot of people are putting much more weight on the human in vitro assays in particular now, especially to replace the animal studies. I think people don't really pay too much attention to immunogenicity in rodent or non-human primate studies for fully human proteins, for example. So there's much more focus on these in vitro tools because we're staying within a fully human system. So as I said, I'll just touch very briefly on in silico immunogenicity assessment. This can be a very useful platform to use. So we have our own in-house tool, and there are a number of other tools available out there. These can be very high throughput and very low cost ways to look at the sequence-based risk of immunogenicity. You can look at potential epitopes that can drive HLA class 1 and HLA class 2 responses for both CD8 and CD4. Usually for biological therapeutics, we're more focused on the CD4 response that provide help to B cells to produce anti-drug antibodies. But for other cell and gene therapies, mRNA, etc., you also have to consider the class 1 response. And our tool, just like lots of other tools, essentially just takes that amino acid sequence of the protein of interest, breaks it down into individual tenmers, so every linear continuous 10 amino acid stretch within that full sequence. And each of those short peptides is then predicted for binding to all of the different HLA that's present within the tool. So we've taken the approach to look at as many HLA as we can. So we don't just focus on supertypes or subtypes and just DR, for example. We try and cover DR, DP, and DQ alleles for class 2, for example, to give us the broadest coverage possible and the broadest idea of the level of risk. We should note that obviously the in silico tools can be overpredictive. They're sort of inherently overpredictive by their nature. And I think that's fine as long as you recognize that and you use them in the right way. But they are overpredictive for a couple of reasons, mainly because of the way we break down that protein sequence. We know that not every tenmer exists when it gets naturally processed and presented within the immune cells, but that's very difficult to predict. So we can't currently really predict that very accurately, but we know not every tenmer will exist. So we're looking at predicting binders that may not be able to bind once they're naturally processed within the immune cells. And we also know that obviously we're not predicting T-cell receptor interaction and T-cell activation here. We're just looking at which bits of my protein are going to bind to HLA, which are then presented to the T cells. We're not asking, answering the question of whether there's going to be any TCR that can recognize that. That's very difficult to predict. I'm not aware anyone can do that currently because of the diversity within the T cell receptor. But we have built in some filters to try and address this. So for antibodies in particular, we can obviously include antibody germlines. So we can focus in on the most non-self bits of the antibody, typically the CDRs or any regions of the FC that you've mutated for ADCC enhancement or silencing or half-life extension. So it really helps us focus in on the highest risk peptides that could be binding to HLA from the protein of interest. But as I said, we do have to acknowledge that they can be throughput, they are low cost, but we're looking at an overrepresentation of what's potentially presented to the T cells. So obviously, to do a much more in-depth, accurate assessment, we can then move to in vitro assays. There's a host of different things we can do with the assays. Just going back to the schematic that I showed a little earlier, we can break down responses in patients that lead to anti-drug antibodies into the innate and adaptive arms of the immune response. I'm mainly going to focus on the adaptive here. I may just touch very briefly on the innate. And then I will give one example of the immunotoxicity assays that we run in particular for cytokine storm prediction. So I'll work my way through some examples across this platform of assays that we typically use. So probably the most important thing that we do is the cell bank. So we spend a lot of time collecting, optimizing this process to make sure we've got very high quality cells. So this is the single most important thing to all of the in vitro assays that we run, is that you have very good high quality cells to start off with. If you don't have high quality cells, no matter how good your assay is, it's not going to perform well and robustly if you have low quality cells. So we actually collect either whole blood or leukopaks, primarily from healthy donors in the areas very close to our labs. So we collect in the London area for our Cambridge, UK labs. And we have a number of sites very close to our Boston labs as well. So within sort of 20 minutes in Boston, and maybe within a couple of hours, we have the cells in our lab in the UK. And that's key. So really minimizing that time from the cells coming from the donor to having the blood and leukopaks in the lab, to isolating the cells and then control rate freezing and storing them in restoring vapor phase nitrogen. So really minimizing and optimizing this process makes a huge difference in the quality of the cells that you have in your bank. So we typically have anything between 500, 1,000 plus. Sometimes it changes quite a lot. We're continually collecting. We've actually switched more recently, as I mentioned, to leukopak collections, because we can get a lot more PBMC from each individual donor, which allows us to do much larger batches of samples. We can do repeat testing. We do much more complex assays. We can increase the number of cells we use in the assay, which is obviously important for things like a T cell response. So we've actually increased the number of vials in our bank, but maybe we collect slightly fewer donors because we're getting more vials from the leukopaks. But obviously, it's important that we HLA type all these donors, and we have a good diverse bank that we're using in all of these assays. So that's obviously something that we do. They're all HLA typed. We run a quality control analysis for all of the PBMC preps to check that we have good recovery, viability post-thaw, and that the cells are functional when we use them in a T cell activation assay, for example. We also have all the ethical aspects covered as well. We have the Human Tissue Authority here in the UK that governs, how we collect, store, and use samples for research use. And then we also collect serum samples from all of the donors as well. So we do have matched serum samples from every donor preparation. So we have used this in some instances to pre-screen donors for pre-existing AAV antibodies, for example, in some of the cell therapy projects that we've run. So that's an additional resource that we also have linked to our bank. So I'll just run through a few examples of different assays that we run with these cells. Most of the assays I'm going to talk about here are using cryopreserved cells. We do also run some of these assays with fresh cells in some settings and some sort of studies where that's more of a requirement for certain types of product. I won't go into that in too much detail today. But all of these assays work really well on the cryopreserved cells. So I'll just start with the cytokine storm assay. So this assay is one of the simpler assays that we have. We refer to it as a cytokine storm risk assay or a PBMC activation assay. We're essentially just looking at very short-term activation of the PBMC. So we typically go to the donors in our bank, maybe 10, 20 donors selected at random. We're not too concerned about HLA alleles here. Usually the sort of responses we're looking at here are more mode of action driven or off-target driven, more through the MOA or off-target interactions than genetic differences with HLA and T cell epitopes, for example. So we're normally collecting and selecting the donors randomly here. So we take 10 or 20 donors from the bank, we expose that mixed population of all the different cell types that we have within the PBMC to the test products and relevant controls. And then just after 24 or 48 hours max, we would harvest the supernatants, and then we can look at the cytokine, chemokine profiles that are produced in response to all those different conditions. The systems that we can go up to 500 plex with the multiplex Luminex systems that we have. Typically, we're in the sort of 8 to 12 plex. That's usually enough. And obviously, we validate this with the sort of immune responses we're looking at and the right controls to make sure we're covering everything as accurately as we can and have a very sensitive assay. And there is a little bit of case study data here. So we're always trying to benchmark these assays with clinical products. So there's a range of antibodies you'll see here that are familiar to most people here, I would imagine. They're widely used in the clinic. Some of them, like alemtuzumab and rituximab, are known to give cytokine infusion-related reactions due to their mode of action. And as you can see, we've just got interferon, IL-1, IL-6, and TNF shown here. And you can see a very similar pattern where we can see differences or increases in most of these cytokines to alemtuzumab and then slightly less to rituximab, but you can still see it's higher than the lower risk controls, like things like cetuximab, for example. And then we have more strong polyclonal controls like anti-CD3 antibodies or anti-CD28 antibodies that are known to give fully blown cytokine storms in patients due to the mode of action. So we've done a lot of work to sort of benchmark these assays to make sure that we're able to detect problematic or risky molecules at these early stages, and we're not really missing anything. And we've got a number of cases that we're not able to share, but there have been a number of studies that we've run where we have highlighted issues in this assay. And while that's not great for that individual program and for that particular client, obviously it's better to work that out now and highlight that now before you're in a patient. And you have, that's going to be much more costly timeline-wise, going back and engineering or changing your molecule, and also reputation-wise with the company. You don't want to see these issues in clinical trials. You want to try and dig them out a little earlier. So that's the kind of only the one immunotoxicity assay, and that's kind of an assay we would recommend for any product, any modality, anything that's being dosed into a patient. That gives you useful information. We do have some people trying to use that assay for dosing prediction. That's a little bit more difficult with sort of cells in a well compared to an IV or a sub-Q dose of a product. But we do see some reasonable correlation with some products, so that's something that we're investigating a little further. I did also touch on the innate immune response. I'm not going to go into that in detail right now. We actually use the same assay, so the cytokine release assay works for innate immune responses as well, short-term cytokine, chemokine release. The main difference is we change the controls that we use, and we're running lots of these innate immune response studies for generics and some biosimilars and some for process changes to support regulatory filings on altered impurity profiles. So if you've changed the process, or if you're trying to develop a generic or a biosimilar, is your impurity profile or your risk of activating the innate immune response the same as the reference drug that's already out there in the clinic? So as I said, the assay is very similar to what I just outlined. It's just the sort of controls and test conditions that we include. But that assay works really well for innate immune response. I'll move more now into the adaptive immune response. So this is more kind of following on from the in silico tool. So as I said, the in silico tool predicts HLA-binding peptides within a protein sequence, but we're not taking into account the natural processing and presentation of those peptides from the protein. So this is where the MAPS assay comes in. I'll come to the case study a little later on, but just to give you a little bit of background on the assay itself. So here we're actually taking monocyte-derived dendritic cells from donors in our bank, from the cryopreserved bank. Here we are selecting them based on their HLA. So we normally primarily select them based on their HLA-DR alleles, but we also keep an eye on their DP and DQ to make sure we've got good broad coverage in the sort of 10, 20 donors that we would pick for these studies. And then what we're doing, we're taking the PBMC, we use automated magnetic bead isolation systems to pull out the monocytes, which we can then differentiate into dendritic cells. And it's these dendritic cells that we treat with the protein of interest. And then we allow them to take up, start to process, and then we activate those dendritic cells with LPS, for example. And then the cells switch from being very efficient at taking up protein to be very efficient at processing and presenting peptides from that protein via HLA receptors on the cell surface. So at this point, we've got very activated dendritic cells. They've upregulated the HLA-DR and other class 2 alleles on their surface, and they're loaded with peptides from internal, from the media, and from the protein of interest as well. So this part we all do in-house, and then at that point we harvest the dendritic cells, and then we actually subcontract the next steps, the immunopurification and the mass spec analysis, to a specialist immunopeptidomics CRO called ImmuneSpec. They're based in Antwerp in Belgium, and they have a really nice optimized process to basically immunopurify the HLA peptide complex for HLA-DR, DP, and DQ. I think they also do class 1 alleles, which I'm not going to talk about today. We just focus on the class 2. But it's a really efficient process to pull out those HLA-binding peptide complexes. And then they have very high-resolution mass spec systems to then identify those peptides that are eluted from the HLA, and then they map back onto the protein sequence of interest. And it's a platform that we've moved to recently, and it's working really well, especially compared to the kind of more traditional approaches that used lower resolution mass spec and maybe focus more on HLA-DR only, and not also the DP and DQ, which we now know are also very important for immunogenicity risk. So I just want to give you a quick snapshot. So this isn't the case study. I'll come to that later on, but this is just a quick snapshot of how the data looks. I appreciate you probably can't see the sequence or any too much detail here, and that's fine. That's not the intention, but it's just to show that obviously we have the heavy chain sequence here. So this is actually with infliximab. When we feed the infliximab antibody to, I think there's just a few donors here with a few different conditions that we've used. And if you see a red bar, that's where we found peptides in that individual donor. So each sort of row is an individual sample here. And then if there's a red line, it means we found a peptide at that location in the sequence. And we've got residues 1 to 80, 81 to 160, etc. throughout the heavy chain. And then the darker the red, the more peptides we found. So the more dense, epitope dense that region is. So you can see there's a region here from around about 80 to 100 or so where we find a number of peptides across that region. So it's a very quick visual way to see where are the hotspots in my protein. And then obviously knowing what that sequence is it self, is it non-self, then that helps you understand what risk that could potentially pose to activate a T cell. And then on the right-hand side, we have a slightly different way of showing this data where we have the CDRs marked up on the sequence, and then we have each sample split into each individual box as you go down the figure, and then we've got the heavy chain and the light chain, and you can see here each individual colored little tiny bar is a peptide that we found. So this is showing each individual peptide. So you can see there's a region across the framework and into the CDR3 in the heavy chain that's much more, that's the high risk area, and I think there's a number of publications and data out there showing this same region coming up. So this is what we did, just sort of set up, optimize, validate the assay. And as I said, it's working really well. It's very robust. The main variety or variation we see in the assay is from the donor HLA. It's very donor HLA driven, as you would imagine. So even though that's much more accurate than an in silico tool, excuse me, we're still not predicting whether that can raise a T cell response. We're still looking at what's presented to the T cell. So to obviously take things a step further, we need to then bring T cells into the assay. And there's a variety of ways of doing that. I've just got a few examples here. One of the assays we use for following up on MAP studies is a peptide screening assay. So we basically take PBMC from a bank, we deplete the CD8s, so we enrich the CD4s, and then we add peptides that we've either designed from in silico studies or from the MAPS assay themselves, or just overlapping peptides across a region or across a whole protein. And then we can test those individual peptides, dose them onto the CD8 depleted PBMCs, and then after a week or so of culture, we can detect the number of activated proliferating T cells with flow cytometry. So we run a lot of high throughput flow cytometry here to give us the throughput to do this. And this works really well. It's quite a nice assay, especially for peptides. It's been heavily optimized for this process with the right controls, which I'll come to in a second. And just to give you an idea of the scale, so if we're taking on the whole project, we can pretty much run in 6 to 8 weeks, we can run in the region of 50 peptides in a 30 donor cohort, which we would usually do for the T cell assays to make sure we're covering all the HLA that are important, but we've also got some T-cell receptor diversity across a number of different HLA and different donors as well. And that sort of 6 to 8 weeks also includes the peptides. So we can include the peptides, synthesize the peptides, and then run the assays when those peptides come in, and then basically correlate that data with MAPS or in silico, whatever we've had previously, to really look at which peptides are immunogenic and where they are in the protein, and then what do we want to do with that data as we go forward. And then just to note, yeah, there's a bit of examples here on the sort of controls we use in these assays. So obviously a lot of people use KLH. Typically, all of our donors would respond to KLH in this particular assay. We use class 2 peptide pools like the CEFTA peptide pool, which is a cluster of viral peptides. I think there's 35 different virus peptides from CMV, EBV, flu, tetanus, and adenovirus, and most of the donors respond. And then we've got individual peptides that represent either memory or naive responses. And we've recently switched to including lots of clinical peptides, things like the GLP-1s, calcitonin, that we're using for some of the generic studies. We're using them to benchmark these assays as well to show that we've got a good proportion of our donors that can raise a naive T-cell response in this particular assay. But one thing I just wanted to make a note on, that obviously the sterility, the purity, solubility of the peptides is key here. So if you don't have a really highly pure, soluble, non-aggregated peptide, you'll see false positives due to something else going on, due to particles or aggregation. You can get non kind of HLA TCR driven responses here in these sort of whole PBMC assays. So that's a really key thing to control, is the quality of the peptides that are included. So that's for peptide screening. We then use a slightly different assay usually for whole protein screening. This is a much more complex assay, as you can see. So much like the MAPS assay, we're isolating monocytes and generating monocyte-derived dendritic cells in pretty much an identical way to the MAPS assay. We're treating them with a protein of interest, and then we activate the cells or mature those dendritic cells. But rather than lysing the cells at this point and immunopurifying the HLA peptide complexes, we're actually going back into the same donor, isolating autologous CD4 T cells, and we add those T cells onto those dendritic cells. So the T cells only see HLA peptide complexes from the protein or peptide that we've dosed the dendritic cells with. And then we typically co-culture the T cells and the DCs for around a week or so. And then in this version of the assay, we do actually re-stimulate them. So we go into the donor for a third time, isolate autologous monocytes, treat them with the same protein or peptide that we used to load the dendritic cells, and then we add those to the T cells. And then we do a sort of 24, 48-hour fluorescence spot just to look for a boosting of those cells. And you can see the spots that we have in the bottom right of this slide. The green spots are interferon gamma secreting T cells, and the red spots are IL-5. So we're looking at Th1 and Th2 T cell responses here. And again, we've validated this assay in-house. So you can see the graph on the right of the slide here. Again, we've taken some antibodies that are known to be immunogenic in the clinic or known to be much less immunogenic in the clinic. So we've got infliximab at one end and bevacizumab at the other end. So you'd expect much fewer patients to raise antibodies against bevacizumab than infliximab, and you can see we see a nice difference in the percentage of donors that we have responding to those two products in this assay. So it's typically less than 5 or 10% the low risk products, and then anything 15, 20 to 30% typically with those higher risk products. And there's also some independent validation of this assay platform. So there was a publication from a few years back from the Roche group. And they've done a lot of screening in our assay, and they were really great and really nice enough to do an independent validation on all of our raw data and all of the different controls they've included, and that's really nicely outlined in the paper. But the pros and cons of these assays, where the sources of variability are, what the accuracy is. I think it was typically around about 80–85% correlation with the clinic, which we were pretty happy with. And there's a few examples of how they in particular use it in their pipeline screening. So that's a really nice paper to look for an independent validation of these sorts of assays. So just before I move to the case study, I just wanted to touch on some of the applications of these assays. So there's obviously a lot of different modalities and sort of ways you can apply these assays. What we see in most cases is that we're using these sorts of studies to support lead selection or risk assessment of a handful of leads. That's usually where they're most powerful, using them very early on, using them to inform your lead selection and optimization. But as I touched on, we're also doing lots of equivalence testing of generics and biosimilars and process changes. So if you've had a process change to a marketed product, has that changed the impurity profiles? What does that mean for an adaptive or an innate immune response? So we've applied a lot of these sorts of platforms in this context. And then the type of products in the lower half of the slide, we've obviously used lots of antibodies and antibody-like molecules and fragments, lots of bispecifics and T-cell engagers and weird and wonderful immune modulators that can give us a headache in the assay sometimes. Sometimes we have to do some pre-work to check that we can basically pull out the influence of the MOA from what's a true immunogenicity response. Some of them are immune suppressors, and then we have to adapt the assays to accommodate that. And we're doing lots of work now with not just biologics, but also vaccines, toxins, cell and gene therapies, and mRNA and LNP, which I'm not going to talk about today. But one thing that the case study that I'm going to jump into now, just to kind of for the last section of the talk, we also apply these tools to investigate adverse events in the clinic or unexpected or unwanted events that we see in the clinic. So that's what I'm going to talk about in the case study. So this was using the MAPS assay, and this is actually for an antibody drug conjugate, so for an ADC. So this particular ADC, a client came to us with the ADC, and they'd been into early clinical trials, and pretty much all patients had developed ADAs against the ADC. I think they found ADAs both against the antibody itself and also against the conjugate part, the payload part. Important to note, the conjugate is non-cytotoxic, small molecule so there's no additional protein added here with the conjugate and the linker. And in this case, the client actually engineered the antibody to add the payload via site-specific conjugation. So I'm not going to talk about that bit too much, and any details obviously are all redacted. But basically, this was taking an antibody that's not immunogenic in the clinic. It's widely used in the clinic as both a naked antibody and as an AD. Generally has very low immunogenicity. So why did we see so much immunogenicity here, which really wasn't expected? So we became involved to then try and see what assays can we use to try and understand what's driving this increase in immunogenicity compared to the naked antibody. So what we did, I'm just going to show data here for the MAPS assay that we've just got in the last few weeks. And essentially what we've done is taken 10 donors. We selected donors to cover major DR, DP, and DQ alleles. And then we tested individually the naked antibody, the modified antibody that had a few point mutations to site-specific conjugate the payload. And then we tested the full ADC. So we're looking at what influence or what impact did those sequence changes have, and then what impact does adding the payload on top of that have. Typical assay that I went through here, followed the process, loaded the dendritic cells, harvest them, immune spec, then did the immunopurification and mass spec analysis. And then we basically looked at the peptides that we found, mapped them back to the heavy chain and the light chain of each antibody and ADC. And then as controls, we also look at the total number of peptides that we found in each sample. We also look at the whole human proteome, which I'm not going to show data for here, but I will just focus in on the albumin peptide profiles, just to check that we haven't had any major impact on the function of the DCs after treatment with the antibody and the ADC. So I'm going to show you some peptide maps again, like I showed for infliximab a little early on. So this is the standard heavy chain, and then we've got the variable domain and the constant domain in two different blue colors here. I've noted on where the CDRs are, the CDR 1, 2, and 3 on the heavy chain. And then as you can see, the red bars underneath for the 10 donors, so each row, if you like, going down, is a different donor. And then essentially we work our way through the sequence. So you can see in the standard heavy chain, so this is the clinical antibody that's widely used, and it's not immunogenic. And as you can see, we don't see too many peptides. Most of them are in the framework and the constant domain. There's only a couple of donors here that are running into one of the CDRs. So this looks pretty low risk, as we expected. There's not too much that would concern us here. When we then compare that to the modified heavy chain antibody, so as you can see, we're seeing the same areas coming up, but we're seeing additional areas coming up as well. So that immediately leapt out. Interestingly, I haven't marked on here where the site-specific point mutations were made, but there are no new epitopes across the regions where those mutations were made. So it's not that we've made a mutation and introduced a neoepitope. That's not the case here. So there's something else going on that I'll come to a bit later. And then the third sample, when we add in the payload on top of that modified heavy chain, we see more of the same. So very similar to the modified heavy chain, but just more again. So there's an additive effect of the mutations and then adding the payload onto that. And then we see a very similar thing for the light chain. So again, this is the standard light chain for the clinical antibody. Very few peptides, very few in the sort of non-self high-risk areas around the CDRs. When we move to the modified light chain, again, we see a bit of an increase, not as pronounced as the heavy chain, but a general increase across the whole sequence. And then this is further amplified when we add in the payload. So very similar kind of result that we see across the heavy and the light chain. So I mentioned earlier, one of the controls that we use is human albumin. So this is a key control to check that we're just not seeing this with every protein that's in the sample. The nice thing about albumin is that, so we don't use any serum in our assays, but obviously the serum replacement factors we have in our media, albumin forms a large component of that. The dendritic cells shouldn't be producing albumin themselves. So the albumin is entering the dendritic cells through exactly the same pathway as the antibody that we're treating into the media on the top of those cells. So it's a really nice control for going through the same process as the test drug in this case. And obviously here we have many more rows because we appreciate you probably can't see all these different rows. So these are the 10 donors and the 3 conditions, the 3 antibody treatments per donor. So if you look at the pattern of responses, it doesn't matter which bit of the sequence you look at, you can see they come in kind of triplicates. So you see very reproducible responses with the donors, especially if you look at the middle section, the bottom of the middle section, you'll see 2 very red areas in particular, and that's in the same donor. So what we see here is the donor variability based on their HLA, but the technical variability is very tight. We see even the same numbers of peptides across the same area. So this gave us a good indication that it's not necessarily every protein that's in there that's interacting with the dendritic cells, that we're seeing a general increase when we add the ADC compared to the naked antibody. And then finally, if we look at that a little bit more numerically, won't spend too much time on this, but yeah, in green we have the standard antibody, the modified antibody in orange, and then in red the modified antibody with the payload. So you can see we're increasing from around just less than 10 to almost 50 peptides per donor. So this is the average number of peptides per donor in the heavy chain on the left, and in the light chain we're going from 5 to 20, 25 peptides per donor. Whereas the albumin peptides, they're pretty much identical across the 3 treatments. So we're around about just over 100 peptides per donor. And then the total peptides that we found in each sample are also pretty stable at 6,000 to 7,000 or so in this particular measure. So it's very specific to the heavy and light chain in this case, which is obviously interesting and pretty unexpected. So yeah, this is data that we've just got very recently. So it's nice that this pattern correlates with what's seen in patients. As I said, we don't see any neoepitopes where the mutations are made, which was interesting, which is kind of what we expected initially. We thought, oh, you've modified a human sequence to do the site-specific conjugation, you've introduced epitopes. That doesn't appear to be the case here. But there is some literature out there to suggest if you aggregate antibodies or if they're polyspecific or charged areas or sticky parts of the antibody, that can change the way that they interact with dendritic cells and other immune cells. So maybe there's other kind of physicochemical properties of the antibody that have been changed by those mutations that are further enhanced when we add the payload. And this is leading to just more of the antibody getting into the dendritic cells. And then we're either creating new epitopes just from the sheer amount of antibody, or there's differences in the processing, or because we're just getting so much more antibody in per dendritic cell, are we just seeing an increase in the sensitivity to detect all those very low level peptides that would normally not be really well represented and present in the antibody without those modifications. So there's a lot of kind of biology, immunology going in here that we don't fully understand, but there are some other cases out there in the literature to show similar things with aggregated antibodies, for example. So yeah, we can obviously do some follow-on studies here to look at the activation of the dendritic cells. Does it change how they're activated in any way? And what does this mean for the T cell response? We're assuming that this is going to lead to an increase in T cell activation if it's associated with an increase in anti-drug antibodies in patients. There must be some way that they're boosting the CD4 T cell response to have that help to the B cells to produce antibodies. So there's a lot more we can do in investigating this in future. So yeah, this really shows that we can use the MAPS assay to really investigate the mechanisms behind clinical immunogenicity. That's not really what we'd like to be doing. We're very happy to do that, but really we want to be doing this during development. So obviously, if we run these assays and generated this data during the development of this particular ADC, we would have highlighted that this, maybe we need to look at alternative approaches here or do something a bit more conservative because there's risk here in increasing the immunogenicity risk. And that's generally our main message is that the earlier we run these sorts of studies, the more powerful they can be and the more they can really reduce that risk of having these issues when you get into a clinical trial. So just a couple of conclusions to wrap up before we get to the questions, and I can see quite a few have come in. So it's important to note that, obviously there's not just the MAPS assay. I touched on some other assays as well. There's lots of other class 1 and class 2, CD4, CD8 assays, potency assays, binding, etc. There's lots of things to consider. I think, and that's another reason for thinking about this as early as possible, so you can really work out what makes sense for that individual molecule, what are the risks based on the MOA, the origin of that modality, the nature of that modality that we're going to be testing. So there's a lot of different ways you can do that. So it really pays to look at this as early as possible and think about what you might need to do, excuse me, and what data might be helpful as you develop that particular drug. And then just finally, in the last slide or two, just to help show how we apply these sorts of different assays and technologies across different modalities, I focus more on sort of what we would do for biologics here. And for lead selection, we do a lot of the cytokine release, the cytokine storm assay, as well as some of the T cell assays to help run a sort of sanity check or help select a lowest risk lead. If we're doing engineering of a biologic, in the bottom left box here, we can obviously bring in silico. MAPS can be very useful for helping to deimmunize proteins and also the peptide screening as well. So there's a host of different ways we can approach those. I touched on a lot of the work that we're doing with biosimilars and generics and process changes. And interestingly, there's a requirement by the regulators now for generic peptides in particular, and generic oligonucleotides. You have to run innate and adaptive studies to support the ANDA filing. So we're learning a lot about how far we can push these assays, how we can qualify, validate them for regulatory filings as well. So that's a rapidly moving area of the field as well. And I won't go into too much detail, but obviously we do a lot of work in the mRNA, LNP. Lonza has a whole mRNA business and a cell and gene therapy business as well. So we do lots of work in that space as well, in a very similar way, screening which mRNA, LNP combinations are likely to give the highest or lowest risk, depending on whether it's a vaccine or a therapeutic. And then looking at things like cell and gene therapy with AAV screening that I touched on a little bit earlier as well. There's different assays and different approaches we would take for each sort of modality and approach. So just finally, before we move to the questions, hopefully that I've shown you that the in vitro assays can really form a critical component of the preclinical risk assessment for immunogenicity. Human immune cells and human immune cell assays are a great way to do this. Exactly how we run these studies really is a case by case, or depending on the type of product and the sort of type of target and disease indication, etc. And we approach different modalities like T cell engagers, CGT, mRNA, peptide, etc. We can approach them all slightly differently on a case by case basis. It's really important you have high quality cells for all of these platforms. And then with the case study, hopefully I've shown that MAPS can form a really nice component of troubleshooting unintended clinical responses, but even better to use those earlier and avoid those problems in the first place is what we typically recommend. So thank you for your attention. Thank you everyone for joining. Just like to acknowledge the US and UK teams that we have here at Lonza that do all the work. Obviously all of this is generated by these guys. We have great lab teams at both sites. And just to thank you again. Apologize for me coughing intermittently, but thank you for your attention, and very happy to move on to the questions.
Thank you so much for your presentation, Dr. Simon. And now we're ready to move on to the questions. The first one is, do you offer these in silico prediction analysis as a service for peptides, therapeutics, and its impurities, and for generics?
So yeah, so we do offer the in silico tool as a service as well. It can have its limitations for the generics and impurities. All of the in silico tools are primary amino acid sequence based. So if there's deletions, insertions, truncations, elongations, etc., we can take those into account if it's a linear amino acid change. But if there's structural changes or post-translational modifications or switches in the L to D enantiomers of the individual amino acids, the in silico tools can't predict what impact they have. So we do sometimes apply those in silico tools to help look at impurities in the generic peptides. But pretty much in every case, there's always at least some peptides that we can't evaluate in silico. So then we need to run the in vitro assays. So we're still working this out. There was a workshop with the CRCG and the FDA a couple of weeks ago, or last month in October, and there's a lot of focus on exactly how we run these adaptive studies to look at the T-cell response, what sort of assays we use, what sort of controls. We're also looking at whether we can use MAPS instead of the in silico. So if we do see hits in a T-cell assay, for example, can we then use MAPS to follow up and see, is this an artifact due to particles or something else, or purity of the impurity, or whether it is genuinely a HLA binder and is being presented to the T cells. So yeah, in silico is limited in that setting at least.
Great. Okay, next question is, do you do peptide cleanup after acid elution in MAPS?
Yes, so this is obviously done at ImmuneSpec, so they handle that part, but yes, there would be some cleanup of the peptides. Not sure exactly how they do it off the top of my head, but yeah, once they elute, there will be some cleanup before it goes on to the mass spec. Yeah.
Okay, next, in your ADC example, while you're testing a non-cytotoxic ADC, do you still assess and confirm DC viability? How does in silico compare to MAPS data for ADC, native mAb versus thiomab?
Yep. So that's a great question. So yeah, we do keep an eye on the viability. Obviously, if we see a reduction in the number of peptides with an ADC, is it because it's just killed all the dendritic cells? That's important to monitor. So we look at the viability of the cells and the activation state of the cells. There's also a step that ImmuneSpec carry out to check that they spot check some samples to check there's similar HLA across that they isolate by Western blot and check. So there's a number of stages that we check that there's no catastrophic things happening from in respect to an individual product. Sorry, can you remind me of the second half of the question?
How does in silico compare to MAPS data for ADC?
Yeah, thank you. So actually the in silico data correlates really well with the MAPS assay. So if we're trying to compare in silico to clinical or in silico to T-cell assays, then we're not really comparing apples with apples. If we compare in silico to MAPS, they're much more direct comparison. And in general, we have our own tool and there's a number of other tools out there, and you'll see that most of them will, everything that you see in MAPS, you've probably also predicted in silico. But there will be some things that are predicted in silico that you don't see in MAPS because of the processing. So there could be some peptides that are predicted to bind to a HLA-DR in silico, but they get cleaved in the middle when they're processed, for example, or they're too long to really fit efficiently within the HLA. So yeah, generally it's a false positive issue with the in silico when we compare it to the MAPS compared to a false negative.
Awesome. Okay. How much DC, how many DCs are needed for each donor for MAPS assay?
Yeah, so that's a great question. So we typically use around about 1 million or slightly less by the time we've taken some out for the phenotype analysis. I think across the industry, that's what usually people are using. I think some people have shown you can use less. I think some people have shown some data from clinical trial from patients as well with the MAPS assays now, because they're much more sensitive. You can get away with lower numbers of cells. We haven't pushed it yet to see how far down we can go. Obviously, with our donors in-house, we don't have a limitation on the numbers of cells. We can use a lot more cells than 1 million if we choose to. So it works really well with 1 million. And most of the data that I've shown there is at or between 800,000 and 1 million dendritic cells.
Thank you. Next question. In your ADC example, how are the MAPS correlated with the DC/T cell assay readouts? Fully expected that question. We don't know yet. So we haven't run those assays.
I'm not sure whether our client would run those assays in-house before we run the map. So that's something we're going to look at to see whether, in a PBMC proliferation assay or a DC T-cell assay, do those differences pull through, or is it more complicated than that? We don't know. And I think I mentioned in one of the slides, we plan to look at the activation of the DCs as well. Is there some kind of difference in how this is as activating the DCs and what role the payload is playing as well? So that I'm not able to talk about the payload, but there could be some complications with the payload mode of action as well in this case. Although we were seeing the differences only with just the non-payload modified antibody as well. So there's a few different things going on that, hopefully we get to investigate and follow up with another webinar in 6 months, maybe with some more information.
Awesome. What types of assays would you recommend if the protein of interest, in parentheses antibody, has an MOA that affects the immune cells?
Yeah, so we're certainly seeing lots of those. It depends on how it affects the immune cells. So normally we would, if it's a T-cell engager or an immune suppressor, normally by switching to a dendritic cell T-cell co-culture assay, we avoid the direct activation or suppression of the T-cells. So usually that works. There are some products, though, some T-cell engagers that inherently stick to the dendritic cells and get carried over. We've seen that in the past. We've also tested some products that bind to dendritic cell receptors that the dendritic cells have, and that's obviously an issue. In those more extreme cases, we run evaluation studies in just a handful of donors to look for this. Is there suppression when we spike in the product with a KLH control, for example, or are we seeing polyclonal activation? We can do all those investigations. If there's nothing we can do to take out that MOA, the backup option is to go to peptides. So we can make peptides either across the whole protein or areas of the protein. There are caveats to peptides, especially if we can't use the MATS assay to help us identify the naturally processed and presented peptides. But obviously, we may be looking at an overrepresentation of risk by testing peptides that don't exist naturally. But that is kind of the last option that we have if we can't take out that MOA influence, is that we just make peptides across the areas of interest and test those individually or as pools.
Thank you. So we have 2 more questions. And speaking of peptides, when you do the peptide assay based on in silico and MAPS data, do you reconfirm that the peptides are not further processed to smaller peptides?
Okay, so what we see, I'm not sure whether I fully understand, apologies if I'm not answering the question directly. What we actually see with the MAPS data is we often see sort of nested peptides across a core epitope region. So we do see kind of the N and C term of that core changing in the MAPS. That also sometimes the in silico data can help us really work out what that core binding epitope is. So we often use the MAPS data and the in silico together to really work out what the core epitope is for us to ultimately test. We don't do anything other than that to really look at whether there's any further processing of the peptides. There may be something that ImmuneSpec does. I could check in with those guys to see if there's anything, but nothing that I'm aware of that we do to check further processing.
Okay, thank you. And then the last question, there are a few questions within this. So I'll read it all, and then I'll read it like question by question. Okay. So in the peaks, when we see the HLA coverage, how do we know which sequences are HLA peptides? I mean, is there any specific sequences for identifying the peptide in the whole protein sequence? Do you do any specific digestion in sample preparation that helps you to identify the peptide sequence?
Okay, so I'm not a mass spec expert, so I might not be the best person to answer this in detail. I could get back to you on this one, but I know there's obviously when you have all the peaks data, you can do you can look for the MHC motif. So there are some tools available at IDB that can really work out what the core epitope is within the peptide that you found. So you can do that in silico, and there's additional tools that can do that. So that's something that ImmuneSpec do as part of the data analysis. When they identify the peptides, they look at what the motif is likely to be and what HLA that's likely to bind to. So there are ways the in silico supports the further kind of interrogation of what's really going on and what HLAs are involved in presenting that peptide. Obviously, with the MAPS assay, when we're pulling out the HLA-DR, DPDQ, there's 2 alleles for, I think there's 6 or 8 altogether. We've got the DRB1, the DRB345, DP, and DQ, and there's 2 alleles for each of those. So there's a lot of sources where each peptide can come from. There were a few donors actually that I forgot to highlight as we went through, that there's very similar patterns across 2 of the donors in the case study. And that they have pretty identical HLA, or they have at least a few common HLA, and we see very similar profiles, and that's something that we see quite often. So yeah, hopefully that's at least partially answering your question. As I said, I'm not a mass spec peaks expert. I don't think there's any specific digestion in the sample prep. Obviously, we're trying to look at what peptides are natively processed and presented from the dendritic cells, and just trying to identify them rather than further processing them. We're trying to keep it as untouched as possible. So I think hopefully that at least answers some of the question.
Great. And we actually just got one more in as you were answering that one. For the MAB or ADC treatment with DC cells, do you have suggestions for the protein concentrations?
Yeah, so I think across the industry, you'll see people using anything between 10 and 100 micrograms per mil. We typically use around about 50 micrograms per mil, which is about 300 nanomolar for a standard IgG1. That's an important consideration that I haven't discussed here, that obviously if we're trying to test head-to-head slightly different molecules that have different sizes or different beads on a string or add-ons to the antibody, it's often important to do the molar concentration. So we're looking at it per sort of molecule concentration rather than weight of protein. So that's something else to consider. And we know that's important for the peptide impurities for the FDA and other regulatory filings, that they request that we test things or normalize things based on a molar concentration rather than a weighted protein. So that's a good question. Generally in the sort of 10 to 100 microgram per mil, you'll see people using in the dendritic cell assays.
Thank you so much. Okay, if we don't have any other questions, we can wrap up. So I just want to check, does anybody else have any final questions? Okay, not seeing anything come through. So thank you again for this insightful presentation, Dr. Smith, and thank you for all the attendees for joining the webinar. The on-demand version of this webinar will be available very soon, and a link will be sent by email to everyone who registered. Feel free to watch this or any of our other on-demand webinars for you, and you can also find those on our YouTube channel. Thanks again, and have a great rest of your day.
Thanks, everyone.